Hi all, good morning to everyone. Today I am going to teach you about cell cycle. So, phases of cell cycle. Two types. One is called interphase and another phase is called as M phase. M phase, which is also called as mitotic phase. Interphase basically divided into three types. One is called G1 phase, S phase, G2 phase. G1 phase, which is also called as gap 1 phase, S phase, which is also called as synthesis phase, G2 phase, which is also called as gap 2 phase. Gap 2 phase. G1 phase. During the G1 phase, cell is continuously grow, but DNA replication does not takes place during the G1 phase. In the S phase, DNA synthesis will be takes place. DNA synthesis will be takes place. And in the G2 phase, what happened? The proteins which are required for the M phase, that proteins was synthesized in the G2 phase, which is called as gap 2 phase, So during the S phase, chromosomal number does not double, only chromosome content will be double. From 2C to 4C, but chromosomal number is always constant, that is 2n. Chromosomal number is always constant, that is 2n. M phase. M phase, which is also called as mitotic phase. M phase, which is called as mitotic phase. We can see mitotic phase in diploid organisms. Human beings are example for diploid organisms. What is mean by haploid cells? Haploid, and what is mean by diploid? Haploid means single set of chromosome is called as haploid and double set of chromosomes is called as diploid condition. So, the clear-cut explanation about interphase. Interphase, it is the first phase of cell cycle. Interphase is the first phase of cell cycle. So, this is called as nucleus. Who discovered nucleus? Nucleus was discovered by the scientist Robert Brown. Robert Brown discovered nucleus in the year of 1831. He discovered nucleus. So this is called as nucleus and this is called as centriole. So nucleus was discovered by Robert Brown in the year of 1831. See, in interphase, we can see nucleus like this. After the interphase, which means after the G2 phase, the next phase, which is called as M phase. During the M phase, actually, this is the decondensed form of chromosome. It is the decondensed form of chromosome, which is called as chromatin. This decondensed form of chromosome is called as chromatin, and this condensed form of chromosome is called as chromosome. The decondensed form is called as chromatin. This chromatin, which is made up of DNA, and this chromosome, which is also called as storehouse of DNA. The chromosome, which is also called as storehouse house of DNA. In this figure, both the chromosomes are identical to each other. Both the chromosomes are identical to each other. Such kind of a chromosomes are called as homologous chromosomes. If you observe the human beings, we can see 23 pairs of homologous chromosomes. Both chromosomes are similar. Such kind of chromosomes are called as homologous chromosomes. Such kind of chromosomes are called as homologous chromosomes. So, if you see interphase is divided into three types: G1 phase, G2 phase, G3 phase. Sorry, G3 phase is not there. G1 phase after S phase, S phase after G2 phase. During the gap 1 phase, cell is continuously to grow, which means cytoplasmic content will be increases in the G1 phase. During the S phase, DNA replication takes place. DNA replication takes place, and also centriole duplication takes place. Centriole plays an important role in the animal division. In animal mitosis, animal cell division, the centriole plays an important role. So this is the structure of DNA. The structure of DNA is double helical structure, which is made up of 4 nitrogenous bases: adenine, thymine, guanine and cytosine. These 4 are 4 nitrogenous bases where DNA molecule is made up of. Okay. After that, G2 phase. In G2 phase, whatever the materials or proteins which are required for M phase, those proteins were synthesized in the G2 phase. This is about a interphase.